My topic is plasmid vectors. Joshua Liderberg coined the term plasmid. Plasmids are extra chromosomal DNA molecules. They are small, circular, and have the ability to replicate autonomously. Replication of plasmid is not under the control of chromosomal DNA. They are mostly found in bacteria. Some of the eukaryotes like yeast and plants also contain plasmids. Their ability to replicate independently makes plasmid a cloning vector in the recombinant DNA technology for transferring and manipulating genes. Many antibiotic resistant genes in bacteria are present in plasmids. The size of plasmid varies from a few base pairs to thousands of base pairs. Plasmids also get transferred from one bacterial cell to another by the process of conjugation. Plasmids carrying a specific gene are introduced into bacterial cells which multiply rapidly, and the required DNA fragment is produced in larger quantities. Plasmids are used to prepare recombinant DNA with the desired gene to transfer genes from one organism to another. This is known as genetic engineering. Plasmids and bacteriophages are frequently used as cloning vectors in DNA recombinant technology. For genetic engineering purposes, plasmids are artificially prepared in the lab. The lab-grown plasmids, which are used as a vector, contain an origin of replication, cloning site, and selection marker. In plasmid structure, a vector element ORI, that is origin of replication, is a DNA sequence where initiation of replication starts. Selectable marker for selecting bacteria containing desired plasmid, for example antibiotic resistant genes and other specific genes. Multiple cloning sites, that is MCS. Recognition sites to insert foreign DNA fragment by using restriction enzymes. A few or single recognition site is preferred to avoid getting several fragments. Promoter region promotes transcription of the target gene to get the desired protein. Primer binding site, the sequence of DNA used as a start point for PCR amplification and sequence verification. DNA is cut at the specific points by using restriction enzymes. It's a molecular scissors which make sticky ends of the DNA. Herbert Boyer and Stanley Norman Cohen together discovered recombinant DNA technology by recombinant DNA segments as desired and inserting them into the bacterial cell to get the desired protein. The desired genes are then inserted by using DNA ligase. The recombinant DNA molecule is then introduced to the host bacterial cell by the process transformation. The recombinant plasmid then multiplies using host DNA polymerase. First plasmid used as a cloning vector was PSC101 of Salmonella typhimurium. They showed that a gene from a frog can be expressed in the bacterial cell. E. coli plasmid is frequently used as a cloning vector. First one is PBR322 vector. So PBR322 is a commonly used cloning vector in E. coli and has tremendous applications in cloning. What is the full form of PBR322? P, that represents plasmid. BR, it's a Bolivar and Rodriguez. 322 is a numerical designation. It was constructed in 1977 in the lab of Herbert Boyer at the University of California in San Francisco. It's a synthetic polymer and was the first artificial plasmid to be constructed and used as a cloning vector. PBR322 is one of the most studied plasmids. It is 4361 base pairs long. It is completely sequenced, which means the whole sequence of PBR322 is known and studied. Its molecular weight is 2.83 into 10 power 6 Daltons. Structure of PBR322. origin of replication, restriction enzyme sites and selectable marker sites. First one is origin of replication. The origin of replication in plasmid PBr322 is known as PMB1. The copy number of this plasmid is 15 to 20. Restriction enzyme sites. Around 40 different restriction sites are present on the genome of PBr322. Almost 11 different restriction sites are present in the region of tetracycline resistance region. In the ampicillin resistant region, 9 restriction sites of different enzymes are present. Some of the known restriction sites are BAM-H1, HIN3, EcoR1, SA1, SA2 and many more. Selectable marker sites. 2 selectable marker sites or antibiotic resistance genes are present on the genome of this plasmid. Ampicillin resistant site, the ampicillin gene codes for beta lactamase, which can be used for screening microorganisms when a foreign DNA is being inserted in the plasmid. Tetracycline resistant site. This gene degrades the antibiotic tetracycline and can be used for screening microorganisms. The antibiotic resistant genes are useful in screening organisms after cloning. Screening of recombinants containing PBr322. Consider that the restriction enzyme BAM-H1 is used to cut the plasmid at that specific position. BAM-H1 lies in the tetracycline resistance region of plasmid PBr322. Now the recombinant molecule of PBr322, which includes the newly inserted DNA molecule, will be sensitive to tetracycline. But this recombinant molecule is resistant to ampicillin, as the ampicillin resistance gene is fully functional. Hence the recombinant cells are plated on the medium containing ampicillin and incubator. The colonies that appear on plates containing a medium with ampicillin or transformed colonies containing cells with the newly inserted DNA molecules. The replica plate technique is used to confirm the transformed colonies. To confirm that the colonies on the ampicillin containing media are transformed colonies, they are plated on the plates containing a medium inclusive of tetracycline. The colonies that do not grow on the medium inclusive of tetracycline are transformed colonies, as the transformed colonies are sensitive to tetracycline. Advantages of PBr322. Due to its manageable size, plasmid PBr322 is widely used as a cloning vector. The presence of 2 antibiotic resistance genes eases the selection process of recombinants. Multiple restriction enzyme sites make the plasmid compatible in many ways. It has high copy number, which is highly favorable in genetic engineering, top of form, bottom of form. Disadvantages. Detection of recombinant cells having the newly inserted DNA molecule is time consuming. Next one is PUC19 vector. PUC plasmids were first developed by Joachim Missing and his co-workers. It's a commonly used cloning vector in the bacteria E. coli. PUC19 is 2686 base pair in length. The molecular weight of the PUC19 vector is 1.75 into 10 power 6 Dalton. It's a small plasmid with a high copy number. It contains the LAC-Z gene and has multiple cloning sites. Hence, it is widely used as a cloning vector. PUC19 plasmid is similar to PBr322 plasmid in structure. PUC19 full form is P represents plasmid, UC represents University of California, 19 is a numerical designation. The structure of PUC19. Structure of PUC19 is a 2686 base pair long plasmid. Origin of replication. The origin of replication of the PUC19 plasmid is derived from PMB1. Multiple cloning sites. There is a short sequence of 2.8 kilobase which contains sites for various restriction enzymes. This increases the number of potential restriction sites available, enabling the production of the desired fragment for cloning. Selectable markers. PUC19 plasmid contains an ampicillin resistance gene which can be used to screen the recombinants. The plasmid also contains the E. coli gene LAC-Z which encodes for beta-galactosidase. This beta-galactosidase hydrolyzes lactose. Restriction sites. PUC19 vector carries a 54 base pair long multiple cloning site polylinker containing 13 different hexanucleotide specific restriction endonucleases sites. Some of the restriction sites are EcoR1, Hint3, BAM-H1 and many more. Screening of PUC19 vector. The blue-white screening method is used for PUC19 vectors. The process of screening is as follows. This method of screening is based on the fact that the blue pigment is formed when beta-galactosidase catalyzes the hydrolysis of synthetic substance known as XGAL. XGAL is 5-bromo-4-chloro-3-indole beta-D-galactopyranoside in the medium. When XGAL is hydrolyzed, it forms galactose and 5-bromo-4-chloro-3-hydroxyindole. The later, the product undergoes dimerization and oxidation. As a result of dimerization and oxidation, a blue pigment is formed. The cells which contain the beta-galactosidase activity form blue colonies, whereas the cells which do not show beta-galactosidase activity form white colonies on the agar medium containing XGAL. The recombinant cells which contain newly inserted DNA fragments lack the beta-galactosidase activity and hence appear white on the agar plates. This method of screening the recombinant cell is the easiest and fastest method. Advantages of PUC19 vector. This is a small cloning vector and has large industrial applications. It has one-step selection process for the recombinants. Hence, it is used on a large scale. It has a high copy number. The presence of many restriction sites makes it suitable for cloning. Difference between PBR322 and PUC19. Both PBR322 and PUC19 are popular plasmids that are used as E. coli cloning vectors. The prefix P in both vector denotes plasmid. They have several restriction sites. In PBR322, this vector is 4,361 base pairs in length. In PUC19, this vector is 2,686 base pairs in length. PBR322, the BLA gene encodes for ampicillin resistance and the tetracycline A gene encodes the tetracycline resistance. The LAC-Z gene encodes for beta-galactosidase. This vector also encodes for ampicillin R. In PBR322, it provides approximately 20 plasmid copies for a cell. So, low copy number. In PUC19, it provides approximately 500 to 700 copies per cell. High copy number. And PBR322, its molecular weight is 2.83 into 10 power 6 Daltons. It is bigger in size than PUC19. In PUC19, its molecular weight is 1.75 into 10 power 6 Daltons. In PBR322, the restriction sites in PBR322 are Hint3, BAM-H1, SAL1, PST1, PUV2, PUV1, CLA1 and EcoR1. In PUC19, the restriction sites Hint3, BAM-H1, SAL1, SMA1, SAC1, KPN1, XBA1, PST1, SPH1 and EcoR1. Thank you.